Welcome everybody. In our last class, we have discussed about cell cycle, different phases of cell cycle. Today we are going to discuss about the checkpoints of cell cycle. So what is actually checkpoints? Checkpoints are actually the controlling process. Checkpoints are actually the controlling process which ensure controlling process which ensure the proper division of a cell, which means actually whether the preparation for the cell division is as per requirement or not. Preparation for the cell division is as per requirement or not, that all the process has to be checked at these points. So there are three basic checkpoints which are noted. Three checkpoints are noted, which are G1, G2 and M checkpoints. First of all, we'll discuss about the first checkpoint is G1 checkpoint. This is the first checkpoint, which is G1 checkpoint. In eukaryotes, it is also known as restricting checkpoints. This G1 checkpoint in eukaryotes, it's also known as restricting checkpoints. The things that has to be checked here, which are as follows. What the things are to be checked? First of all, whether the DNA is damaged or not. So, first of all, damage of DNA has to be checked, whether the DNA is damaged or not. Because DNA has to be replicated at S phase, so it is very much essential to check whether the DNA is damaged or not. If the DNA damage occurs, the cycle halts at G1. That means it might go cellular arrest. If the DNA damage occurs, the cell might go cellular arrest. So first of all, damage of DNA has to be checked. Next, the number 2 which are to be checked is the enzymes, proteins and ATP. All these things are properly there or not has to be checked, because this S phase is involved the replication of DNA and the enzymes, protein and ATP, which all are going to be required at this S phase for DNA replication. So these things has to be checked. Thirdly, activation of cyclin. Activation of cyclin. So what is cyclin? It's a group of protein. The cyclin is a group of protein that initiate cell division. This cyclin is a group of protein which initiate cell division. This activity of CDK is also checked. CDK means cyclin dependent kinase. CDK or cyclin dependent kinase is responsible for initiation of transcription, which ensure that now the cell is ready to enter to the S phase. This CDK or cyclin dependent kinase is responsible for initiation of transcription that ensures that now the cell is ready to enter to the S phase. It is the point the cell becomes committed to entering the cell cycle. The cell becomes committed to entering the cell cycle. So, all these things has to be checked during G1 checkpoint. Next we see the G2 checkpoints. In G2 checkpoints, actually what happened? what are the things that has to be checked at G2 checkpoints? First of all, in G2 checkpoints, whether the DNA has properly replicated or not. That means completeness of DNA replication. First of all, what has to be checked? Completeness of DNA replication. Whether the DNA has properly replicated or not. And then, has the cell sufficiently accumulated protein, enzymes, or ATP, particularly the tubulin protein, that is the accumulation of enzymes, protein, ATP, particularly the tubulin protein. This tubulin protein is responsible for the formation of spindle fiber. This tubulin protein is responsible for the formation of spindle fiber. So, the synthesis of tubulin protein has to be checked at this point. If everything goes normal, the cell now moves to M phase for division. If all these things comes normal, the cell is now ready for go to the M phase or mitotic phase. In M phase, the third checkpoint is there, that is M checkpoint. M checkpoint is the third one, which is also known as spindle checkpoint. This is the third checkpoint, which is also known as the spindle checkpoints, which actually occur at the metaphase stage. Actually occur the metaphase stage of the M stage. Why this is called spindle checkpoints? Because at this stage, the spindle fiber has to attach to the kinetochore of the chromosome, and the replicated chromosomes, the DNA replication occurs at the S phase. And now, if the chromosomes, if I draw a chromosome like that, there is the kinetochore, and these are the spindle fibers. The attachment of spindle fiber to the kinetochore has to be checked, whether the spindle fiber is properly attached to the kinetochore of chromosome or not. That has to be checked, because the attachment ultimately leads to the proper separation of chromosome. This separation of the chromosome is occur through the attachment of the spindle fiber at the kinetochore. These sister chromatids will be separated to the different poles, the poles of a cell. So that has to be changed. So spindle checkpoints is actually covers the area whether the spindle fiber is attached to the kinetochore or not. So attachment of spindle to kinetochore. Attachment of spindle to kinetochore is the main focus for M checkpoints. So if we overall see what's actually happening, so we can say something like that. If we recall or summarize the checkpoints, what we can say? First of all, G1 checkpoints. What is happening at G1 checkpoints? Integrity of DNA. Integrity of integrity of DNA has to be assessed at G1. Number 2, G2 checkpoints. What actually happened at G2 checkpoints? Proper chromosome duplication has to be assessed. Proper chromosome duplication has to be assessed. And at M checkpoints, what is actually checked? Whether the spindle fiber is properly attached to the kinetochore. So, attachment of spindle. So, these three checkpoints, the main functions of these three checkpoints are something like that. At G1, integrity of DNA, proper chromosome duplication has to be assessed at G2, and attachment of spindle to the kinetochore has to be assessed at M phase. The molecules called cyclin and cyclin-dependent kinase are responsible for regulation of the cell cycle. These acts as regulators for cell cycle. So, now if we can see the initial structure of the checkpoints, you can see the first checkpoint is G1 checkpoints, the next one is G2, and finally the M checkpoints. So this is all about checkpoints of cell cycle. In the next class, we'll discuss on metaphase cell division. Till then, see you. For testing your understanding of this lesson and more videos, log on to www.tubelessons.net.